Where's the magic? This one. Ah, okay. So thanks for the nice introduction. So I want to talk about how we stress or how we deal with documentation and the reproducibility of our data analysis at the Functional Genomics Center in Zurich. First, who we are. So the Functional Genomics Center is a joint research and technology center of 2 universities in Switzerland. It's rather old. It's founded more than 15 years ago. It's an open technology platform, so we are kind of flexible which technologies we are supporting. We are running more than nearly 200 research projects per year. So a lot of projects are running in parallel, and our user base is also contain more than 2,000 researchers of both universities. We have several branches right now in this Functional Genomics Center. There's a sequencing unit. Microarrays are a bit outdated now, but we have also proteomics, metabolomics, and biophysical analysis. And so I'm working in this sequencing department, and in the sequencing department, there is a genome informatics group, so kind of an embedded bioinformatics unit. And I'm a member of this unit. A typical NGS workflow, also in, of course, another core labs, is that the bench biologists doing the lab stuff, the wet lab stuff, the library preparation, sequences, creating raw data, and then there's a workflow necessary to do the QC and the analysis. So that's then our job, and to get interpretable results. And in our case, we developed our own tool to run all these workflows. It's called Sushi, developed I think 4 years ago, and it was published last year in BMC Bioinformatics. So Sushi stands for supporting users for shell script integration. So it's a simple graphical user interface workflow manager. The key concept is that it creates shell scripts or job scripts, and then these job scripts are submitted then to the computing cluster. It manages all the meta information, so sample to relation from the samples to the files, which library prep was used, all the QC measurements, which Phil also showed, are managed in Sushi, and it's documenting automatically all the analysis steps which are used in Sushi. So right now there are more than 50 different apps integrated. So an app means it's mainly a wrapper of an available tool combined to create reports or to create output files. And it's very easy to add a new application to Sushi. So our purpose, our idea to develop Sushi was that bioinformaticians in our own group can use it, but also our users of the sequencing service. So that's for both, for biologists and for bioinformaticians. The typical data analysis process in Sushi is that you are starting a Sushi application based on the input data set. So based on the files and the data information, only a couple of applications are available. Then you feed it with your parameters to configure your job. This will create this job script, the shell script, parameter file, and also the next meta information file, a new data set. And as a second step, it's simply submitted to the grid via the grid engine, creates result files, data files, or result report, and also the log files. So this is an automatic process. So for the reproducibility and for documentation, the key features of this tool is that it creates a job script, which is a shell script, which can also be executed outside of Sushi later to get exactly the same results. It's creating the log files, as I mentioned before already, which are then kept. It creates for the configuration all the parameters which were used to run this application, and of course it creates also a metadata file. So and all the things are then stored, of course, in the Sushi database, but also in the long-term storage together with the final result files and with the reports. So for every job, this information are always available and kind of forever to document what was done. So besides Sushi, we take it very serious how to manage the software tools and in our environment. And to do that, we are using modules. Some technical details, we are using Lmod, which is a Lua-based approach for these modules, which are available on any Unix system. And the modules are enabling us to add or to remove directories to the path environment variable. With this simple from we can control the analysis tool version which was used. So I have a simple script here from a start job created by Sushi, and there's only one line needed if you use these modules, where you add then to the shell script automatically by Sushi. It's added, but which star version was used, which sum tools version. Here's some pre-processing stuff from FlexBar and Shromatic and so on. So so you are really, with these modules, you can really version you have all the different versions available, and even in a couple of years we could run with this old version of star, which will be outdated in a couple of years. We could rerun it with the same version. We could also run it, of course, with a new version, but it will be still available, the old version, with these modules. The modules are not working for different R packages, so we are also using a lot of R scripts in our environment, in our pipelines, which are triggered then by Sushi. But we are collecting them all in our own R package called Easy Run, which is at least on the version control on GitHub. And for customized analyses, which are not part of this Easy Run package, we try to create our own R packages, separate R packages to ensure the documentation and versioning and put them on GitHub. And what's important for at least to keep track which package versions were used from all the bioconductor packages and CRAN packages, we always keep the session information. We also put it in the reports if we create results from R. And in most of the cases, we also store the entire session, the entire R session, to be able to get back. Then for the reproducibility of the data, I think it's also very important to have a systematic approach to store all the references and annotations in a sequencing unit. So we were inspired by this Illumina iGenomes and used exactly the same structure. We just extended it at the end here for the genes, that we have different versions for the gene models. So if it's ensemble-based, the providers ensemble, I think they're providing every 3 months a new release for the gene models for every species. So then we don't need to create the whole branch, a new version, but just a new, based on the gene models, a new version in the structure. And same for the variants, if there's a new version of dbSNP and so on for the species available. So, and we keep all the old versions, we document which version, which reference plus the gene model version was used for a certain application. Then we can always go back to the same reference, because I think using a new reference can have a big influence on the final results. That's why it's important, I think, for the reproducibility to have also the old ones still available. So in summary, we try to document everything in our workflows, mainly via Sushi. We have a systematic approach for references, reference repository. We are controlling the software versions via modules. But I have to admit, I mean, in, let's say, 10 years, it will be difficult to create exactly the same environment, what we use now. And also in a different lab, it could be a bit painful to have exactly the same versions running. So, but I'm still convinced that with a full documentation, that might be enough for a research lab. But of course, if it's if it's a clinical setup, you might probably need a different solution. In that case, we would consider certainly to use this container technologies like this Docker images, really to freeze an environment together with the configuration. And maybe another point for the discussion, I mean, we are also developing a lot of Shiny apps, which are then interactive based on the reports based on the results we are generating. And I mean, this is a difficult task then to document when a user is creating some specific plots for the publication to how to track exactly how the parameters were selected and so on. So in best case, it would create also an R script as an output, which can then rerun outside of Shiny to recreate exactly the same plot. But we are working on this, not existing right now. So finally, I want to acknowledge my team, from the Functional Genomics Center, the Genome Informatics team, mainly mentioning Masa, who was the main developer of Sushi. So thanks for your attention, and I think now we can start with the discussion.